I'll start. So yeah, when you think about designing a data set for experimental, um, for predicting reactivity based on experimental data, I think the first thing you need to think about is what you want to do with your model. So what is exactly the task you want your model to do? And when you think about that is what is the chemical space you want your model to make predictions on? And this question is pretty important to design your data set intentionally to have a model that's going to be able to do that. And when you think about that, you have to put that into regard to experimental constraints and what kind of data you can acquire, how much data you can acquire, and how that balances your goals of having a model predicting on a large domain of applicability. And when you think about those two parameters, you can start designing a data set and designing the experiments you want to do to build your models and perform your experiments. I think two recent projects in C-CAS that have been thinking about that are the project we led in the Reisman group on predicting the site selectivity on complex molecules and how you think about building a data set of small molecules to predict on more complex substrates. And then the Dole group has been working on thinking about how you select pairs of substrates for one given reaction to try to predict which pairs of substrates are going to give high amounts of products on a whole set of alkyl and aryl bromides. And so the first task you need to do is to define your task. And when you want to refine a little bit of what that means, is when you have a reaction, you have to choose what is the space you're going to model. So are you going to model substrates? Are you going to model reaction conditions? Are you going to model just the ligands? And then you have to think about what is the target you want to model. So there's a lot of experimental outcomes you can measure, which reaction rates, selectivities, yield, with the caveat of those different targets can be more or less noisy, and then you have to take that into account when you design your data sets. And then finally, I think you have to think about what is your goal. If your goal is to just have a model that predicts which pairs of substrates are going to react, and maybe you just want to do a classification model, or if you want to think about more how you learn about mechanistic implications of a ligand design for selectivity, then you probably want a regression model. And once you define clearly what you want to do, you have to put that into regard to experimental constraints. The first thing I think that is important is how many experiments you're going to do. And that will tell you a little bit about how much dimensions of your modeling space you can screen. Now, when you vary one or two or three types of conditions, usually the data set size you want to predict on will increase as a power law of the number of conditions you're going to vary. So you're pretty limited into that, especially if you do just traditional batch experiments. You can go a bit further in terms of parameters if you use high throughput. And then depending on the target you're looking at, you might have to make concessions on the chemical space you want to explore. So when you think about data set design, in practice, usually you start with the library of substrate, ligands, or conditions, and what you have as a virtual space before you start your experiments is either the reactants that are commercially available, or the compounds that you think that are synthetically accessible, with the caveat that the synthetical accessibility is not always a very clear measure. And once you enumerated your virtual library, you're going to start with some kind of processing. So if you're thinking about a reaction, maybe you want to process all the reactants that you know are incompatible with the reaction, or you can do pre-screening of a functional group compatibility. And for that, I think the Glorious group has a lot of experience and a lot of references that are useful to think about that. And then once you do the filtering, you have a smaller set of reactant or compounds. You need to describe them, so numerize them to be able to do your modeling. That's what we usually call a molecular representation, and we could talk about hours of what molecular representation you want to choose. Usually what you need to balance here is a trade-off between the cost of computing those descriptors. If you use DFTs, probably, and you have a very large virtual library, it might be expensive. And then you have to think about what chemical species you want to describe. So you can think about describing reactants, but you could also think about describing intermediates in your reaction that you think are relevant for the chemistry, or transition states. The idea behind that is that as chemists, we often think that if we give to the model or parameterize the model with a species that is more relevant to the reactivity, we might get better modeling results. In practice, this is not always true, but that's a hypothesis you can make. And once you describe your chemical space, then you can do a first selection. Depending on the target of your modeling, you can use a clustering based selection, which aims at selecting a diverse set of reagents or reaction in your chemical space, or you can try to select reactants or compounds that reflect a specific part of your chemical space. And then usually what I'm going to show here, that I think a good way of thinking about reaction dataset design is to have an iterative design. So when you perform new experiments, then you can inform selections to iterate. So this is this iteration process is really called active learning. I think active learning is a pretty wide term that is not always very well defined. I think a general definition is an iterative query to label new samples. And one domain where it's used a lot is reaction optimization. And then in this case, the query is used to improve the yield or the enantiomeric excess, for example, of your reaction. But in our case, I think we think more about, when we think about dataset designs for building good models of reactivity, the target of the query is to improve the model performance. So how the new experiments we're going to perform are going to improve the prediction of our model. And so I'm going to illustrate that in the case of the recent paper we published, where the task we thought was interesting for our models would be to predict the site selectivity on complex molecules, but these molecules are difficult to access. They are hard to characterize. So if you could make a model that predicts where you can oxidize them, in this case, so we're looking at CH oxidation reaction, it would be very useful for synthetic planning, for example. And the data we can access easily are small molecules that are commercially available, easy to characterize, but the prediction of site selectivity on these molecules is not very interesting because we could just perform the reaction. So the question is how we go from small molecules to complex molecule, and how can we be sure that a model would predict well on a target of interest. So the first thing we did is we start with the first step, which is defining our task that we did, and then we enumerated the library of reactants we can have. So that's an example of data we extracted from the literature with reactants on which you can perform CH oxidation, and that's an example of a target of interest. And then we featureize them. I'm not going to describe exactly all the different featureization we did, but in this case, we thought that simple descriptors that were relevant to that chemistry, which are BDE, the charge of the carbon and the hydrogen of the site, of the reactive sites, and also the steric constraint at those sites were the most interesting. And then when you do that, you can featureize all your CH sites on your training set, which are the small molecules here in blue, and also the big molecules, which are the complex molecules in gray. And what I show here are the target sites on that specific target. And as you can see, you have some small molecules that are very far away from what we want to predict, and you have some sites where we have very low representativity. So the question is, how do we select the blue points here that allows us to make the best prediction on the target, which are the star, and the red point being the site that is effectively reactive. So to do that, we first designed baseline of how you could do an iterative dataset design, which do not include model information, but one is scaffold similarity. So you could think about what is a good training molecule for predicting on this target. And the first thing that comes to mind would be having small molecules that have scaffolds that are similar to that complex target, or having a small molecule where you have the site of the reactivity that are matching. And if you do that, so you can see here the space in gray of the possible molecule we could test, and then the stars are the sites of our complex target. And what the animation I'm going to show you is an animation where you're going to see appearing the different molecules, the acquisition function we choose acquires, and how that reflects how we screen this chemical space and how that affects the prediction of the model. So here what you see is if for scaffold similarity, step by step, we get more molecules in our data set, and the precision of our model prediction are refined, though at the end of those 16 training, the model does not predict the accurate site, which is the site here. But you could see that site similarity, we kind of space that is qualitatively representative of what we're looking for. Another example here would be, this is exactly the same, would be the site similarity. And so you have in pink here the molecule acquired by site similarity. So basically just taking the molecule that have the most similarity in terms of CH description. And you'll see that we select different molecules, and the prediction are refined more quickly and give the good prediction at the end of the 16. So what we show here is that using different acquisition methods of small molecules, on the left the scaffold similarity, on the right the CH similarity, we get different training set and different predictions. So it's difficult to compare exactly in this subspace how the site is, the different CH sites are sampled. But you could see when you look at the structure of the molecule that the scaffold similarity prefers cycles, which have more overlap with the complex molecule, and the site similarity prefers long alkyl chains. One of the pro of those acquisition baseline is that they're pretty easy to implement. But the cons, as you can see, is that there's a reasonable amount of redundancy in the information you give to your model. For example, here, you have a lot of cycles that probably give you redundant information. And here on the right side for CH similarity, you have a lot of alkyl chains that give you probably the same information. So what we thought about doing is, instead of doing those acquisition functions that do not depend on what we learn, we could implement some active learning where we use the information that is learned by the model at each step to select the new compound. So for this example, you could see that if you had a model that predict both reactivity of your CH site and uncertainty, then you could try to choose small molecules that are representative of the sites that have the larger product of reactivity and uncertainty. For example, the right site is predicted reactive, but really not uncertain. So it's not useful to learn on that site, whereas this one on the left is reactive, but also very uncertain. So you want to have a molecule that looks like this. And so we do that. We could have similar acquisition function, and you could see that you kind of have something in between where you have both cycles and those alkyl chain that are probably both important for reactivity prediction, and we get the good accuracy, the good site selectivity in few steps. So in terms of how that shows up in terms of prediction and number of experiments realized, you could compare that to the random baseline, which would be an acquisition function that just selects a random molecule. In this case, you would need around 90 samples to get to a model that predicts accurately the good site. But if you use the scaffold similarity, what we saw it's about with 16, we're not exactly at accuracy. And here you need an average like 30 samples. If you use a site similarity of active learning, you need much less molecules to do good prediction. So what you could also add, but I'm not going to show it here, to your active learning is constraint on which molecule you would like. So for example, maybe you prefer smaller molecules, and you could add a constraint in your acquisition function to add smaller molecule because they would be easier to characterize, and so you can think about processing more data if you sample smaller molecules. And you can be creative if you have other ideas. It's pretty easy to modify acquisition functions. So in conclusion, I think I tried to explain on this specific example how we define a task, and then we thought about how we could think about how to reduce the number of experiments to make reduced activity prediction on complex molecules feasible with a few amounts of commercially available reactions using active learning. I'd like to thank the Reisman group, our collaborators in C-CAS, Professor Anat Milo and Jonathan, that helped us on this project, C-CAS, and the NSF and Caltech for funding, and you for your attention.